papers, peer-reviewed journals. At this time, I pass the presentation over to our presenter, Dr. Shah. Sergey, go ahead.
This is introduction. This presentation will be about vibrational spectroscopy for pharmaceutical applications. In the first few slides, I will give you very basic information what FTIR spectroscopy is, and review application for FTIR spectroscopy, and then we will switch to run vibrational spectroscopy, and also I will show you applications of Raman vibrational spectroscopy. Very basic. So that's all molecules vibrate, and complex vibration of molecules can be decomposed on simpler vibrations. This is an example of a water molecule. And vibration of this molecule can be decomposed to asymmetric, symmetric vibration, and also in a scissoring vibration. Typical vibration frequencies of these molecules, it's in about 10 to the 14, 10 to the 13 hertz. In spectroscopy, we use notation in hertz for the vibration frequencies. We use so-called wave numbers, which frequency in hertz divided by the speed of light, and do calculations. You figure out that frequency of molecules is a line in range of 4,000 to 400 wave numbers. So the question is how we deal with spectroscopy. Well, if molecules have a dipole moment, and if dipole moment changes during vibration, molecule will absorb radiation at a frequency of this vibration. So previous slide, we know that molecules vibrate from 4,000 to 400 hertz. If dipole moment change, vibration molecule will absorb light at that frequency. So 1,000 to 400 wave numbers, infrared light, that invisible infrared light. We can measure how much molecule will absorb, so get information about molecule vibration. Spectroscopy, we are not dealing with no molecules, we are dealing with the samples, how typically experiment is done. So we have a sample, we light will be absorbed by sample, and we are measuring how much light goes to the sample for transmission. So this example of water molecule. So model is simple, so spectrum is also simple. Here are only couple bands in the spectrum. One band is around 1,600 wave numbers, is related to the vibrational molecule, and broadband around 3,300 wave numbers. It actually consists of two bands, and this bands are related to symmetric and asymmetric stretching vibration. So that's very simple molecule, gives a simple spectrum. Now switch to the complex molecule. So example of hexals, you can more bands appeared, and each specific band in the spectrum can be assigned also the specific molecular vibration, like CH3 stretching, carbonyl vibration, and so on, or can be assigned to the vibration of a bigger molecule chunk, like a backbone vibrations. So you see, simple molecule have a simple spectra, complex molecule have complex spectrum, and each have an individual spectrum. So that's why this infrared spectroscopy is called like fingerprint spectroscopy. So what are general applications for spectroscopy? First, chemical composition. So if we can get spectrum, we can identify chemical composition because each molecule has a specific spectrum. Opposite, if we have a if we know molecular structure, we can calculate spectrum of the molecule. Well, we can learn about interaction between molecules, because in different environments, so the spectrum will change slightly, and we can figure out environment effect on the molecular behavior. Information about molecular conformation. So different formats have a different spectrum. Polymorphism is important for pharmaceutical application. So we can get it from infrared spectroscopy. Different crystals and different polymers show spectra. Learn about orientation of molecular segments. So if we stretch Palmer fields or orient molecules like liquid crystal molecules, so we can figure out how good is the orientation of the molecule by measuring infrared spectrum at different polarizations. So let's a few examples of application of infrared spectroscopy for pharmaceutical industry. And the basic one is identification of the samples for quality control. It's shown here few spectra of all drugs. All spectra are very clear different. So basically, if you have material would like to have identified materials, what kind of drugs you use, or you would do quality control of incoming raw materials, you can use infrared spectroscopy. That next slide shows the excipients. Again, excipients shows different excipients, shows you different spectra. We have a mixture of drug and excipients, so it allows you to do library search or multi-component search, so you can software can figure out not only a component, but also identify the mixtures and figure out what is inside the mixture. For plus excipients and software, if you measure spectrum of such tablets, so you can identify of drugs and also identify the excipients used. So infrared spectroscopy can be used not just for qualitative analysis, like for identification purposes. It's also very used for quantitative analysis. Example of a spectrum of methadone in water. And when changing concentration of our sample, so infrared band intensity goes higher and higher and higher. And the well-known Lambert that high absorbance is proportional to the concentration sample and also to the path length. And flow can be used for the quality analysis of a sample. Example of concentration profile. So what you do, you measure some of the several samples with known concentrations, and then make a calibration curve. After that, you can measure concentration of unknown concentrations of that sample. So it's another popular application of vibrational spectroscopy, infrared spectroscopy for in the pharmaceutical industry. Nowadays, you might know, for those who work in pharma, so there's a lot of pharma now switching to biopharma. So instead of work with small molecules, we work with proteins and figure out how the proteins can be used for drugs. Well, for those who are not familiar much with proteins, give you very basic stuff. What protein is a sequence of amino acids. The primary structure of a protein is amino acid sequence, can be identified using mass spec. So also make it mass spec, but that's not the topic of this discussion. Secondary structure. So in secondary structure, well, secondary structure is related to helix content or beta sheet content of the protein and also random coil. So proteins consist of alpha secondary structure consists of alpha helixes, beta sheets, or some random parts. So this part of the protein of secondary structure can be identified by infrared spectroscopy. Structure, so that's a more complex arrangement of segments in the proteins, identified from NMR X-ray. Again, can do an X-ray, but we will discuss this time only application for secondary structure. So that slide shows the typical spectrum of protein, and if you look on the spectrum of many proteins, they very look alike. So big two bands, intense bands around 1650 and 1550 wave numbers. It's called amide 1 and amide 2. The most important band is band amide 1, that's in 16 to 1700 wave numbers. That is extremely sensitive to the secondary structure. So all analysis based on this band is important. This is an example. If you have a completely random coil structure, that band appears as 1645 wave numbers. We have most alpha helix structure, that band appears as 1652. If it's beta sheets, that's the band 1680 and 1630 wave numbers. So if we have a combination of alpha helix, beta sheet, and random coils, so that we can use infrared spectroscopy to identify proportion of this part of the molecules. Easy, but the problem is how to get a good quality spectrum of protein. And the problem is water. You need to measure protein in a natural environment on the buffer. Buffer is water buffer. And with the water itself have huge absorbance around 1600 wave numbers, 60 wave numbers. That showed you in introductory slide. Water vapor, which is atmospheric water vapor, have a very sharp bands and also located around 1650 wave numbers. The problem is if you have a low concentration of protein in a buffer, so you need to be very careful to subtract water contribution and atmospheric water contribution. And to be very careful with this is an example of how good spectra can be collected. And in order to collect it properly, you need to a big cell with a control cell, and that should be temperature control as good as 0.1 degrees Celsius. Temperature should be very powerful and should be also stable within 7 centimeters. Otherwise, you will not subtract some water. Sealed, because if it's not sealed, then you will see contribution of atmospheric water, and again, you will not see spectrum of protein because it will be under absorption of atmospheric water vapor. So how do you deal with this? So Brook or a system called check, which is actually consists of two components: transmission cell and AT accessory. That's transmission cell, and it's for protein stability, quantification of structural changes, the nature, secondary structure examination. So we have its temperature control within 0.1 degrees Kelvin, and also password stability within few nanometers. So it's also sealed. This is application that can be used. Second accessory, so it's called the AT accessory. We developed this accessory in collaboration with Harvard Scientific. It's used to do temperature ramp of proteins, so aggregation processes, the melting points of proteins, brain proteins. Let's give you a few examples how this accessory can be used. One example is of concentration of proteins. That is how I can do it. So let's measure good spectrum of proteins. Did a broker, we created a model, concentration versus absorbed versus concentrations for the set of you can see the higher concentration, more absorbance. This model, based on Lambert-Beer law, you can use this model from broker to when you measure the spectrum. After you measuring, you run this model, and that you can get concentration of a protein. And it's the good things about this is the calibration range of 1 milligram per milliliter to 100 milligram per milliliter, and also it not depends on the type of protein you use. So any protein can be used with this model, and you can get concentration of this protein. So the point to get good quality spectrum of protein in with broker check system, you can do it. So typically measurement time takes about less than half a minute. This model. So this is the things which is we can do concentration. Second, structure. It's easy to figure out the secondary structure of protein. I selected over 43 proteins with known secondary structure from X-ray analysis, and use a partial least square modeling to be a model of in alpha helix and 4 beta sheets content. And you can see it here. It's shown on this slide. Separation range from alpha helix is 0 to 75%, or for the beta sheets 0 to 48%. And prediction, you can see it looks pretty high, 5.54%. And what is this error? Well, that's because all the data we used are based on X-ray diffraction methods, which have about 5% discrepancy, so error. So the infrared can be better than a primary method as with X-ray. But if you would like to see a relevant in an alpha helix or beta structure, so infrared spectroscopy will be much more precise. This is example how you can do it. So typically, for those who work with proteins in the pharmaceutical industry, one of people need to figure out stress effect on a protein. So what we did here, this is an example how we can use infrared spectroscopy to solve this issue. What we did, we have prepared a set of several proteins and several relations of the same protein, and applied different stress and measure all these proteins before and after stress in using a transmission cell. It's about 100 seconds equivalent time, 4-wave number resolution, and this is a spectrum shows you spectrum antibody protein after different stress applied. Looks like pretty similar. Subtract stress minus unstressed, you will see the big changes. What the changes came from? Well, let's see one band becomes negative. Negative means less loss of negative sheet structure. If one band goes up, means increase of intermolecular beta sheets. So you can quantitatively compare different stresses. So what to do? We calculated stress minus unstressed spectrum protein, and area the different spectrum and take a ratio to the area of an unstressed protein. So basically, bigger area on a different spectrum means more difference between stressed and unstressed protein. Less area, like from sample number 6, you see less influence of stress on the protein structure. It's just in the graph, what is the difference? The change of infrared spectrum in percentage for different proteins at different stresses. Is obviously you can see number 2 is have more difference, and number 6 is less. So formulation number 6 is a high state compared with number 2. Example how you can use infrared spectroscopy for this type of work. So things I have to discuss. So that's where before we're talking about regular infrared spectroscopy. Now, if you have small samples, can you do the measurement of smaller samples? You can. What you need to do is you need a microscope, which allows you to measure samples as small as a few microns. The application for such measurements is of particle analysis of the concentration in the product, variety of coatings, analysis. So this if you have layers in the films or your competitor packaging material, you can figure out how many layers and so on, and do use of heterogenic structures. A few examples of this. Pharmaceutical tablet is inclusion, so that's all departments or big pharmaceutical companies have a sort of forensic science division, which is they need to figure out what's going on with their product if something happens. This is an example of a bad product, pharmaceutical tablets with inclusion, and you need to figure out what's going on. So fix the slit in a sample holder and use a so-called animated total reflectance spectroscopy. That's the sampling technique. Apply spectrum for 10 seconds per each spot. For the resolution, this is how does it look acquired spectra, and show how look spectra. Yeah, in inclusion spectra it shows in like blue. They're pretty different from on the rest of the sample, so it means what we can run the spectra against in library. So we have a library which contains about 26,000 spectra, and obviously we see a very good match that this yellow spectrum of yellow inclusion is very close to the spectrum of magnesium stearate. So that's for information for those who are doing formulation. You need to figure out how happens this magnesium stearate was found in one of these tablets. So just for the solids, how about liquids? If you have a particle in a liquid, so you have to collect spectrum, you need to catch particle. Well, it's not if it's floating, so but what you can do, you can just filter your solution through the mesh filter, and the particles will stay in the filter, on the surface of the filter, and you can basically easy see it on the filter surface, and then put that filter under infrared microscope and do the measurements. This is an example small particle 25 by 25 micron. As you did here, this is how they look under the microscope. So you use infrared microscope. It's just not only infrared; it's also visible, so you can find particle and then take a measurement. Example: take fine particle, take an infrared spectrum of this particle, run against in library. That's another example. That's another good match. It's same, so on. So you can use microscopy not only for small particles, but you also can do chemical image. So you can basically what chemical image is. So you measure spectrum or all spectrum, like in X and Y direction, and profile the surface of the samples that give you distribution of chemicals on this sample. Example of such is about where's the cancerous tissue on the slide. So what is here? Pathologists give us the size of tissues might have a cancer. So measurement using focal ray detector. So that's a resolution about 10 microns per pixel. The first things we measure different points and characteristics spectral features to generate chemical images. This is an example, so you can figure out how the protein is distributed on this tissue. So by calculating area under 1720 to 1480 wave numbers, we do the same things to figure out the distribution of collagen or the carbohydrates on the cancerous tissue, which is that we know that happens in the region of 3300 to 3180 wave numbers. So as you can see here, we can differentiate where the cancerous tissue, where the carbohydrates, where the proteins, and we also can combine image, which just gives distribution of all these components on this slide. This is part of a very basic software which is comes with a FTIR microscope, so that you can generate infrared spectra, multiple infrared spectra, and they can generate images. Now, before I talk about infrared spectroscopy, now we switch to the Raman. Well, Raman is all related to the vibration of the molecule. When molecule vibrates and the polarization of molecule changes, so you can the light, reflected light, will also change frequency. So our experiment is done. So with a laser sample and measure scattered signal. Scattering we only measure scattering at different radiation wavelengths. So say for example, it's called Raman spectrum. This is in Raman. Well, in terms of band position, we are very close. Of course, band intensity will be different because the effect of infrared spectrum depends on the changes in the dipole moment, and Raman spectrum depends on the polarization changes. So technique is better, depends on application. For example, C double bond is very weak in infrared but very strong in Raman. For example, pure oxygen or nitrogen not show spectra in infrared because we don't have any this molecule doesn't have dipole moment, but oxygen and nitrogen can be seen in the Raman spectroscopy. So it's basically that depending on the chemical structure, you can use or Raman or infrared, or you can use both if you have completely no information about the sample. Is a spectroscopy is become popular because it's non-destructive analysis. It's pretty easy to use. You go look for the vials, for the packaging materials, no sample preparation. It's all advantages, but disadvantages if your sample fluoresces, so you not get a Raman spectrum at all. So the biggest enemy of Raman is fluorescence, which is about 1,000 times stronger than Raman signal. So if your sample fluoresces, so have now other choices to switch to infrared. Application of Raman applications is different polymorphs. So different polymorphs use different Raman spectra, different forms. So here, the preference of also have accessory allow you to put as this like well plates, so put different samples and automatically measure all the well plates. So then this we have a software which is automatically run all this well plate and do identifications and quantitative analysis using the PLS model you developed. Example of relations of such applications. So yeah, that's everything is automatic. So you just simply load the accessory, and it's do the measurement in the background, do all the calculation, and shows you results. This we have in infrared, so you can do the same with in Raman. So again, if you have some inclusions in your product or on the particles or something which is unwanted inclusion, you need to identify it. So you can do it infrared or you can use a Raman. So you can go through the files and you can see you don't need to open it. Example of spectrum of the particle you need to identify. This is Raman spectrum, and we do a library search from Raman natural fiber library, and you can see it identifies the cotton. You can also do mapping of it, so to figure out the distribution of chemical, how homogeneous distribution of active ingredient in the tablet, and also what is other inactive or other ingredients in the tablets. That's an example of painkiller tablets. So you can see here there is spectra as a caffeine, paracetamol, acetyl salicylic acid, and we can see the plot distribution of all these components on tablets. Another with Raman, so you cannot do that with but only with Raman. So you can do depth profiling. So how it's done? So you can focus laser lights on the surface, and then using depth profiling, going deep and deep and deeper under the surface, and get Raman signal under the surface. So this is an example of in vitro depth profiling of human skin. It is summary here, and we use 32 excitation laser. The key, as you can see here, so initially concentration is go down at the skin, and then at some point at 23 micron it starts to rapidly increase. So that's how it looks depth profiling on the skin. Okay, about very interesting applications. That's what I showed you. It's only maybe a few percent what you can do with infrared and Raman. Of course, there is much broader application for this. But what instrumentations you can use for this application? Let's overview what broker can, what kind of instruments are available from broker. This simple one is in AI is called Alpha. It's a bench small instruments. Small doesn't mean it's best, so signal to noise is excellent for this. Spectrum is 7500 to 375 wave number resolution. And you can do even kinetics with this 1 scans per second with this instrument. This is for the analytical QC control lab or serial identification lab. So next is we call the Tensor 2 FTR instrument. It also can be extended to NIR, so it can cover NIR mid-IR spectrum range. So that's have a higher spectrum resolution. So also have rapid scan option, which is allow you to do 20 spectra per second. And also you can use different sources, and you can exchange detectors here, use a more sensitive detector if you need it. And next step, that's a true research instrument. It's called Vertex series, Vertex 80, and also there's a 70V and Vertex V. 80V standing for the vacuum, so that's vacuum, non-vacuum version of the instrument. So you have a vacuum, so you do not care about contribution of atmospheric water vapor and CO2 because will not see it under the vacuum. So of course, spectral ranges of these spectrometers can be extended from UV to far infrared. Have a very fast scan options with Vertex A. You can do 87 spectra per second. If you have repetitive measurements, so you can repeat your experiment. You can do even from nanosecond time resolution. You can set several internal detectors, external detectors, also sources as true, flexible, true research instruments. This is very popular for those who is doing research in both academia and in the industry. You can add microscope to the side of the instrument. If you already have an instrument, an FTIR instrument, you could add microscope to the side. This is an infrared microscope. So you get different objectives, gradient angle, ATR, have a very high lateral resolutions, high sensitivity, and it comes with a software for 3D data processing and evaluations. You also can add four-ray array detector, which will allow you to image sample very fast. So I have a stand-on microscope. The microscope doesn't need a FTIR spectrometer on the side. It's self-contained in that microscope. It has all the components, including source, detector, interferometer, to collect spectral or infrared images of a sample. The only things what you need for this, you need to connect to the PC. So it's software for the wizard. Even unexperienced user can easily learn to operate this within half an hour, just this, working on the system. Instrument, we have a stand-on Raman spectrometer called multi-Ram, and then you can see the spectrum range from 100 to 70 wave numbers, point wave number resolution, and for multi-Ram we use a 1064 millimeter laser, and we can do the second laser option of 795 nanometers. There's a bunch of accessories available for multi-Ram, like ADS, high flux spinning, temperature control, the spin, low temperature, high temperature. There is a lot of different accessories. If you need more information, just let us know. And next one, let's see, the newest one, it's updated version of a microscope that we use for fast Raman imaging or mapping. 100 spectra per second for the very fast image of a surface of a tablet or whatever you want to do chemical images. Comes with also high resolution options with 1.5 wave numbers. It also can be combined with FT-RAM and multi-RAM systems, so it gives you more chances to get Raman spectrum, because we mentioned Raman is very sensitive to fluorescence. This is why you can use several lasers on a Raman system. You sample fluorescence at one excitation wavelength, you might not fluoresce in another one. For this reason, that laser system are very popular in the Raman, and the Centera 2 microscope can combine several lasers. And that's what kind of lasers are available on the system and what will be the spectral range from different lasers. finally, we have a handheld Raman. It's called Bravo. So it's basically just a very small portable Raman spectrometer. So that's for you can use for the raw material identifications, get Raman signal. But it's self-contained, so you can point you want to measure, and it's automatically defined how long does it take to measure good quality spectrum. Do automatically a library search, and it basically shows you if it's how good is the match with the library spectrum, or and also can transfer spectrum to standard software, which is called Opus or optical user software. So you use all these standard pre-treatment routines with that spectrum, like spectrum subtraction, multiplication, and so on, if you need to. But typically you don't need to. You just use the Bravo itself. You need to connect even to the PC. And for the pharmaceutical, important application is also 21 CFR 11 certified. Also work on the CMP certifications, and also build wave number calibrations. So all instruments available from Bruker. I'm here an application scientist, so I'm developing a new application. So, and also very interesting to see what you're doing. So if you have, if you can share information with us, what kind of application you deal with, so we can maybe talk with you offline, and maybe I can just visit you to see what you're doing, just to learn from you, and to help you, to show you what we have in terms of applications, and maybe that allow you to solve problems which you have quick and efficient. We'd like to finish, and I can be glad to answer to all the questions you have.
Excellent presentation. Lots of great information, Sergey. Thank you. We're going to allow questions during this particular session. For those that have questions, if you can again just use that Q&A box, and then we can certainly address those questions and provide you with answers very quickly. We did get a couple early on, so I'll go ahead and start with the ones that came through earlier. One of the questions, Sergey, was can the Centaur 2 be used with the fiber optics probe?
Yes, Centaur 2 can be used for fiber optics probes. So you can attach fiber optics to the side of the spectrometer. It's basically two fibers. One is fibers for the laser, goes into the sample, and the return fiber, which is carry Raman signal back to the spectrometer. And use a bunch of Raman probes like for the reaction monitoring, or measure big samples and so on. Okay.
Great. Another question here. What is the spectral resolution for FTIR and Raman microscopes?
Well, it's diffraction limited systems. So on FTIR, so your diffraction limit is 2 to 10 microns, depends on the wavelength. So it's about 4,000 wave numbers. Your spot size is 2 microns. That's 1,000 wave numbers, roughly about 10 microns. This is how we do the measurement for Raman, with dispersive Raman, with full excitation. So you can go with small samples as small as 1 micron.
Okay. Another question. What would be the quickest acquisition time for Raman spectrum with FT-Raman multi-RAM?
Raman multi-RAM, so that's you talked about maybe a few spectra per second, depends on resolution. With dispersive system, you have a much higher acquisition rate, so that you can go to like 6 milliseconds per spectrum.
Probably the similar question then, how fast is the mapping for the Centaur 2? Probably.
That's 6 milliseconds per spectrum. This is showed on one of the slides.
Great. Another question here, can the Bravo be used with the Opus software?
Yes. Brower can be used, and we also can basically that the brower can be connected to the PC through Wi-Fi, so it automatically sends spectra after acquisition from brower to the Spectroscopy software.
And another question: does Bruker offer any process analytical technologies for online monitoring?
We have a AI division. We basically all doing online process monitoring, and for NIR spectrometers, but also you can do is infrared using fiber optics infrared and also fiber optics Raman if you need to. The question is also which technique is best depends on your application. So we need to talk to you, discuss what you're doing and what will be the best and price technique for that application.
Okay. Another question here for the FTIR microscope on foreign particle matters, when to use the ATR versus the reflection or transmission mode?
I would say the ATR probably the first thing, right? If it's easy and not distracting the samples. Reference, if works, if you don't want to touch sample and apply any pressure, the reflectance, that spectrum depends on how good and how bad the surface of your sample. So if it's a rough surface, you might not get good signal. Transmitters require relatively thin samples, so you need to have slice samples to have it about maybe a few microns thick. So again, you spend time on sample prep compared with ATR. My choice will be ATR, reflectance, and transmittance.
Okay, great, thank you. Another question here. Which technique, Raman or IR, is more suitable for the chemical identification?
The techniques works well. It depends on the sample. Depends on the problem. So if you don't want to open container, so that Raman will be the best technique. If your sample is fluoride, so you no way you can use Raman, you need to switch to IR. So once again, it all depends on the sample. So and you have any specific sample to try here at demo lab. So we are open to run your samples for feasibility studies, and I'm doing a lot for different customers. We do not charge for this. So send us your samples in the sample submission form, and we can different instruments and tell you what will be best for you. Or you can come to us. We are in Billerica, Massachusetts. It's about 20 miles north from Boston. So that's you can bring your samples here, and then you see all our equipment. Plus, we can give you a tour of all the Bruker facilities, including the mass spec, and so on.
Great. I just got another question. Have you done any biological applications with Terra 2, and what would be the dynamic range of the system?
Yes, we are doing some biological, not Bruker itself, but our customers doing all the biological, imaging of the tissue imaging. So that's the big area of research. So we use Terra 2 to get the pathologist to figure out where the pathology is. So we do this. The question is about dynamic range. I don't know what exactly that means.
Yeah, either we could probably answer that question offline unless the person asking would like to expand it again. You asked what would be the dynamic range of the system. If you just give Sergey a little more to that question, we'll be happy to answer it further. Another question that came in the meantime was, how does Bruker's Raman system handle rough surfaces for mapping?
Well, that is software allows you to basically how it's done. So if you have a rough surface, of course, you need to focus it every time when you move from one point to another point. And the software basically, when you do mapping, you you software with automatically focus to the surface. So basically, if you have rough surface, so we'll take care about this. So you'll refocus every time when you do the measurement.
No. The question here, is ID testing feasible with an Aqueous DP with other technique?
As I said, yes, that's identification. Yes, you can use Bravo to do for the vials or for other, and you can use any so it depends on a sample. So that's what techniques are available for identification.
Here, the question on the dynamic range, we'll address this offline with the person who asked. Are there any other questions from anyone else coming through? Otherwise, we will go ahead and end this session. We certainly thank you all for your time this afternoon. If you're interested in any of our other webinars that are coming up or have taken place already, we offer a multitude of downloadable recordings for webinars that have already taken place. If you visit us at bruker-webinars.com, we also launched a brand new dedicated pharma site for Bruker. It's loaded with all pharma, so it's not just the IR, it's the mass spec, it includes nano, other divisions, products that are also applicable in the pharma industry. And if you go to bruker-pharma.com, there we will find all kinds of great educational content as well that may help you with your research and your needs. In addition, for those who have participated and those that have registered, we do intend to send you a link to the recording of this session and also the presentation material for further use for you all. So thank you again, and certainly thank you for your time. What a great presentation, and everyone have a great afternoon and a great weekend. Thank you.
Thank you. Bye-bye.